Let's go to Brock Reeve now, the CEO of IVIVA Medical. He's been involved in biotech investments and sees the medical challenges and clinical need for kidney repair. Brock, can you join us?
Yeah, and thank you very much for having me all the way from Boston, or I guess Madrid gets the prize for the long distance travel here. But since he's developing his own airplanes, and he doesn't get credit for that. Yeah, so I'm CEO of IVIVA Medical, which is a startup company in the organ repair space. And I'm also, for the past 15 years, I've been the executive director of the Stem Cell Institute at Harvard. And so let me just give you a little context first on regenerative medicine and the environment out of which IVIVA comes, and say sort of why I think now the time to build a company like that. People have heard about stem cells for 15, 20 years now, but I just remind you that the field has really accelerated in the last 5+ years with, developments such that have enabled us really to understand how to wire and how to program cells. That shows up in a few different ways. One is the Nobel Prize that was won in 2012 by a pair of scientists who showed how you could take a mature cell, turn it into an earlier state, and then reprogram it. And the other was that the Nobel this year to Jennifer Doudna and Emmanuel Charpentier for CRISPR, for gene editing. You should talk to them about purse power, Donna. But but anyway, so now we have a quick, effective, relatively cheap way to put genes in and out of cells, and can combine that with how to tell cells what to do. So what we're doing is developing control over cell fate. What that then lets us do is develop a path for curative therapies for when things go wrong in the body, when you lose capability, injury, disease, et cetera. So far, pharmaceuticals mostly deal with symptoms, symptomatic treatment. The promise of regenerative medicine, stem cells, gene therapy, biomaterials, et cetera, are actually to develop curative therapies. And stem cell science has a bunch of spin-offs. If some of you may say, well, it's just, wacky early stage science, I'd point out that several years ago in the immuno-oncology, in the cancer space, several companies like Juno, Kite, et cetera, became billion-dollar companies in what's known as the CAR T space. These are engineered T cells. You take out of someone an engineered blood cell, fix it, put it back into the body, and cure someone of their leukemia, for example, a curative therapy. There are now cell therapies that are in the clinic for eye repair, both for cornea and retina. There's cell therapies in the clinic for diabetes, for Parkinson's disease, gene-edited stem cells for sickle cell anemia, et cetera. And the technology also has spin-offs. For example, a decade ago, we funded a project looking at using messenger RNA to reprogram a cell. And then that project worked. We then realized, wait a minute, that can be a way of delivering therapeutics. And then they realized, wait a minute, that can be delivering a vaccine. That project became the IP basis for what's now Moderna, which many of you hear about as sort of in the whole COVID vaccine race. So stem cell science and these tools have all sorts of downstream effects as well. So the next stage of development is not only saying, how do you fix individual cells, like when, in diabetes, the disease crashes down on an islet cell, but how do you repair an organ, a complex combination of cells in a complex architecture? Mm-hmm. So IVIVA Medical was started by a colleague of mine at Mass General Hospital. And a couple of years ago, we did a seed round, and we're doing another seed round right now to take the company to the next stage of developing essentially a kidney replacement. So it's a combination of thin film biomaterials where you print channels on either side, which then you can then load with what I call cells of interest. In other words, you could put blood cell on one side and kidney cells on the other, so you can essentially replace the filtering and the reabsorption function of the kidney. And you stack them up, and you put it all together, and then you can have eventually a human scale device to replace the need for transplant or dialysis. Why is that a problem? It's because so far, dialysis hasn't changed over the last 50 years. It really hasn't. And it's a miserable quality of life. And transplants don't solve the kidney transplant problem. In the US, there are 500,000 people on dialysis a year. There are 100,000 people on the kidney transplant wait list, and only 20,000 get them each year. So just huge demand and huge unmet medical need. So the promise of this technique, if it works, is that you can essentially get someone off dialysis and ultimately have a replacement for a transplant, and without requiring a pump, without requiring dialysate, and essentially providing essentially native function. In all honesty, it won't provide 100% of function, but the kidney is a redundant organ. And all you need, obviously, you all know you start with 2, but you only need 1 to survive. And even in any single kidney, if you get about 15% of function, you can get off dialysis. So it's unlike someone with a heart. If you give someone a new heart, you need 100% function so it doesn't electrically misfire, that sort of thing. So there is, a margin. We don't have to have the perfect natural replacement. And the other thing, and the reason we need to do this combination of techniques with stem cell science, biomaterials, and 3D printing is because the kidney, you can't grow a kidney by itself. It's too complex in architecture. A liver has a stem cell, certain organ, simple organs have, or the pancreas is made up of these beta islet cells. You can grow those cells, but the kidney is a very complex structure architecturally as well as biologically. And so this sort of combination of, biological engineering takes advantage of some of these new tools, and that's why we think now is the time to do that. It's also an interesting platform technology. For example, in this era of COVID, people are wondering, how does the virus act, et cetera. I mentioned that in the kidney example, we look at blood cells on one side and kidneys on the other, this membrane and these channels. You can do the same thing, put air on one side and blood on the other to replicate lung function. So right now, we're in a project funded by a foundation to look at putting lung cells on one side, airway epithelial cells, blood on the other, and see not only can we model gas exchange, but what happens when you introduce a virus and what happens when you don't. And when you introduce the virus, and then what happens, and then you can think of, introducing a pharmaceutical or vaccine to say, how can you cure it? So essentially a 3D model of a lung to understand how the disease happens, but also what a potential, therapeutic intervention would look like.
When you're preparing this kidney, my ignorance on the subject, is it still inside the subject's body, or is it outside the body when it's being repaired?
There'll be, no, that's a good question. And there'll be 2 steps. So the idea is first to do the outside the body, the extracorporeal, if you want to sound fancy about it. So that way you don't have to, deal with, because that'll be a lower regulatory hurdle from the FDA perspective, because you don't have to prove it'll be, fine inside the body for X period of time. So the idea is we go to the clinic would be you first do an extracorporeal version and then you do an implant version, much like you do a kidney implant today, and just hook it up to the blood supply and the urinary supply. And where we are so far, so our first seed round, we showed we could have very simple device, a 2-layer device that we hooked up to a pig model and said that it showed that it could perfuse and create a filtrate. The outcome of this next round is to go from a 2-layer to a 10-layer device that we would fully functionally test in vitro and also in large animals, and the pig would be the animal of choice, and to have enough data to go to the FDA to say what would a clinical trial look like. And then for humans, we probably need to scale that device from 10 layers to probably 30 layers.
And so you're still several stages away.
Yeah.
Wow.
Fascinating.
Yeah, impressive.
Yeah, so it's somewhat different from what, what we talked about this afternoon, but, but this is a problem that, just hasn't been able to be addressed by, solutions so far. And mechanical devices, are bound to foul, synthetic membranes foul. So we think that the combination of the scientific know-how from stem cell science, knowledge of cell populations, knowledge of biocompatible materials, now's the time to combine all those into a solution set.
So do you think they're going to be able to grow like any organ in the body to replace eventually?
No, because, for example, this organ, I said you hook it up to a blood supply in the urine, but it doesn't have to be electrically conductive like the heart, right? So you couldn't use this technology to recreate a heart.
Okay.
But you could create what they call epithelial, or so you could, for example, use this approach for the intestine. You could use it for the pancreas. We have a project funded for by Diabetes Foundation, applying this to the pancreas, because you need to have blood supply to your islets and your pancreas. Or you could imagine lung, although the lung also has a physical movement too.
What about like an eye?
No, but you don't need it for the eye, because the eye right now, for example, in retinal cell therapy, if you lose sight, like macular degeneration, that sort of thing, people are putting cells on a thin membrane, and the blood supply there, vascularizes really quickly, and it's really thin. So it doesn't have the same scale problem. So what we're trying to do with this technology is solve large organ problems like the pancreas. Whereas the eye, you don't need that as a solution. You can put either individual cells without a biomaterial or cells on a scaffold, but a very thin scaffold. So you don't need the same level of complexity.
Brock, does it take a pretty specific and specialized investor to want to be involved in your project, or is it more broad-based audience?
I think it's broad-based, and Motion can comment as well. It does take specific investors who can look at the risk, et cetera, but it also takes people who are interested in impact, in the sense, of having an impact on a major disease that can't otherwise be cured. So impact investors are very interested in companies like this and others, because they say, wait a minute, you can have we're motivated by the disease and the condition, and if you can solve it or help move the needle to solving it, you could, they're not only there are huge financial returns, but also health economic returns, as well as psychosocial returns.
What is to add to Brock's point, VC biotech has become an important asset class in and of itself. It's an important sector of technology that gives investors exposure to a lot of things they might not have trouble with prior. But groups like Brock's and the Harvard Stem Cell Institute do a lot of the hard work by incubating these companies, working with them, mentoring them, and before they take investment onto that stage. So Brock has been a leader in this regenerative medicines sector for over 15 years now, and has really developed it. So thanks for all your work, making our job much easier.
I'm not sure we take all the risks, but we can de-risk some of it.
Well, Brock, at the end of the day, when you're successful, will doctors be trained in this procedure, or will there be a series of clinics across the country that can offer this service? How do you proceed?
That's a great question, because many medical devices have failed because people never thought about how does the surgeon actually, who's the customer? How does the surgeon actually deal with this thing, right? In this case, actually, our founding scientist and CSO, Chief Scientific Officer, is a surgeon at Mass General, and we do have a surgeon on our SIB as well, who's head of transplant at Mass General, our Scientific Advisory Board, sorry. And so the idea is, you'd follow the exact same procedure as you would do for a kidney transplant. So any transplant surgeon who's doing kidney transplants would know what to do.